takes place at the synapse, what some of the important structures are. And we're going to move on to compare fast versus slow types of synapses. Okay, fast versus slow synapse. Okay. So there are two types or two speeds of a synapse. There's a fast synapse and a slow synapse. We'll talk about the fast response of the synapse first. This is called a channel-linked receptor or an ionotropic receptor. And this is basically where there's a receptor in the membrane. So we drew a scenario just now where we talked about a generic receptor. We didn't say what type of receptor that was. Okay, so there are different types of receptors. If it were an ionotropic receptor, this would be a ligand-gated channel, a ligand-gated channel. And all that means is that this is a ligand. A neurotransmitter is the same thing as a ligand. And there's a space for it to bind on this receptor. And when this neurotransmitter binds to this receptor, this opens up that channel and we have free flow of ion movement. And that's going to bring about the cellular response. Okay, I'll say that again because students tend to get confused at this point. We talked about a receptor on the cell, and we talked about bringing about some type of cellular response. Now, there are different ways for different receptors to bring about different cellular responses. The first type we'll look at is a fast response, and this is using an ionotropic receptor, which is also called a ligand-gated channel. And ligand means that this receptor is also a channel. All right, so when some type of neurotransmitter, which is acting as a ligand, binds onto this receptor, the receptor then opens. So the receptor is the channel, essentially. When this channel opens, we have the movement of ions, and that will lead to a change in the membrane potential very rapidly. So we will rapidly depolarize this membrane, and we would spark another action potential. After the signal ceases or after the neurotransmitter leaves, right, if the neurotransmitter dissociates from this spot, the channel will then close again. Okay, I hope that's clear. So we have a ligand binding to a receptor. The receptor is also acting as a channel. Once this binds, this channel opens, ions are flowing here, and that will bring about the cellular response. When this ligand dissociates off, this channel closes again. And the difference between this ligand being bound or being free or dissociated off has to do with its affinity to the receptor, right? How much it likes to be around the receptor, which can have a whole bunch of different effects. Okay. All right, so that was your ionotropic. Let's talk about the G protein couple receptor. This is the slow response. This is called the second messenger system. So the fast response system is going to give you an instant change in the membrane potential. It's going to give you that instant cellular response. It's going to be much faster. Why is it faster? Because the receptor is also acting as a channel. We can get things done quickly. There's no middleman. With the G protein couple receptor, this is also called a metabotropic receptor. This is a much slower process, simply because we have quite a few different structures to pass this signal to. So the first option here is that a neurotransmitter binds to this metabotropic receptor, again, which is the slow version of this response, and it's coupled to another protein, right? You can think of this protein as being one of the middlemen or a messenger in this system. And this protein has three different complexes. It has an alpha complex, a beta complex, and a gamma complex. The alpha complex will dissociate off. It will bind to another enzyme, which is somewhere else on the membrane, and that will trigger a whole host of different responses in the cell. So we'll produce some other messengers, which are usually cyclic AMP. We'll talk about that later on, but basically second messengers are other molecules that are released by turning this enzyme on, and they can go into the cell and bring about a bunch of different responses. So in a way, The second messenger system, the G protein coupled receptor, is a mechanism to really amplify the response of one neurotransmitter binding to one receptor. Yeah, I'll say that again. So one neurotransmitter binds to one metabotropic receptor, and we get a whole host of activities that bring about the production of second messenger molecules, which can go into the cell and bring about an amplified response. Okay, so although it's slower, this is going to amplify the response. The G protein coupled system is found in many of our physiological processes, much like the sodium potassium ATPase. It's found in bacteria, it's found in humans, it's found in a lot of other species because it's a very efficient system. Okay, one protein coupled to, or one receptor rather, coupled to a protein can bring about an amplified response later on down the cell. Okay. The second option for this G protein coupled receptor is that there is a slow response, and this is a direct coupling. So this is going to activate this protein subunit, which will bind to another channel somewhere else in the membrane, and this will either open or close this channel. So we can do one of two things. We can activate an enzyme that secretes some type of second messenger molecule, or we can activate another ion channel which opens or closes, and that will bring about a different cellular response. Any questions on the G protein coupled receptors? Any questions? All right, so this is called metabotropic, and this is a much slower response, but again, much more amplified. Okay. Okay, so when we're speaking about graded potentials here, this is depending on whether ligand-gated channels are open or closed. So whenever these channels are open, the membrane potential changes depending on what ions we're moving. If we're moving sodium and calcium, we will depolarize the cell, and this is called an excitatory postsynaptic potential. Okay, an excitatory postsynaptic potential. Why? Because sodium and calcium are positively charged ions, and they're higher in concentration outside the cell. So if you open up a sodium or calcium channel, you will influence sodium or calcium rushing into the cell. That would depolarize the cell, and that would be an excitatory postsynaptic potential. Okay, and all that EPSP means is that we are exciting the cell, we're depolarizing it. On the contrary, if you open up potassium channels or chloride channels, chloride is negatively charged, so if chloride enters the cell, inside the cell becomes more negative. That's going to hyperpolarize the cell. Potassium, although it's positively charged, if you open up a potassium channel, potassium is going to leave the cell. That will also make the inside of the cell more negative. So opening up potassium channels or chloride channels will both have the same effect. You will have more negative charge on the inside of the cell, hence you will hyperpolarize the cell, and these are called inhibitory postsynaptic potentials because these will be decreasing the positive charge or increasing the negative charge in the cell. Okay, any questions on these? So basically the type of channel that we open is going to determine whether we have an excitatory postsynaptic potential or an inhibitory postsynaptic potential. And to sort of make that make a little bit more sense, let's go back to this image. So let's say this channel here was a sodium channel. We have a neurotransmitter binding to this metabotropic receptor. We have the alpha subunit changing its shape. It's going to dissociate off. It's going to bind to this channel. That will open up this channel. If this was a sodium channel, what would happen? Sodium would do what? Sodium would rush in, depolarize this cell. We would have an action potential, right? We would have the inside of the cell depolarizing, becoming more positive. Let's say this were a chloride channel. What would happen?
Inhibitory.
Inhibitory, we bring more negative charge into the cell, we hyperpolarize the cell, we make it hard to fire an action potential. That would be an inhibitory postsynaptic potential. Okay. Ready. So postsynaptic potentials can change the membrane potential in response to receptor neurotransmitter binding. So depending on what type of receptor we found, right, depending on what type of channel it was, what type of neurotransmitter we had, we would either excite this second cell or we would inhibit this second cell. Now, excitatory postsynaptic potentials, the most common neurotransmitter is glutamate. And what that means is that when we have glutamate as the neurotransmitter, we typically open up what type of channels? What type of channels are excitatory? Sodium and calcium. So if we have glutamate as the neurotransmitter, we will likely open up sodium and calcium channels. If we have GABA as the neurotransmitter, this is inhibitory. GABA will likely trigger the opening of chloride and potassium channels. And these will hyperpolarize the membrane and be an inhibitory postsynaptic potential or inhibit the cell. Okay. Okay. So again, just to reiterate this, an excitatory postsynaptic potential is going to produce an action potential just in the way that we described, either bringing more sodium in or bringing more calcium in and depolarizing that membrane. An excitatory postsynaptic potential will allow us to fire another action potential. An inhibitory postsynaptic potential is less likely to produce an action potential. In fact, it's going to make it very hard to fire an action potential. It's going to hyperpolarize that membrane. It's going to drop the membrane potential, right, much more negative than rest, and that is going to make it hard to fire an action potential. Inhibitory postsynaptic potentials are also important in stabilizing the membrane potential. So for example, if the membrane potential was very positive and you wanted to bring it back to rest, you can fire an inhibitory postsynaptic potential, and that would take you from being too positive, make it more negative, and you would get right back down to rest. Okay, so in that way, inhibitory postsynaptic potentials can also help stabilizing the membrane potential, bringing it back down to rest.
How does that work? Like hand in hand with the sodium potassium pump?
So the sodium potassium pump is still churning in the background, still keeping those gradients constant. It's not necessarily affecting this.
For the stabilization?
For the stabilization, that's also going to help in bringing back the membrane potential. Okay, any other questions? Any other questions? So we're wrapping up. Okay, so in this image, we're comparing a fast response mechanism and a slow response mechanism here. So on the left, this is our ionotropic receptor. This is your fast response. Again, your neurotransmitter is a ligand that binds to this receptor. This receptor is also acting as a channel. It opens up. Depending on what type of ion movement we have, we're going to either depolarize this cell or hyperpolarize this cell. So this channel can be a variation of different things. Okay. Likewise, in our slow response, we have the receptor coupled to a protein, a G protein. That protein then changing its shape. The alpha subunit of that protein activates another enzyme. We're not going to be too focused about what the names of these enzymes are just yet, but it activates another enzyme downstream in the membrane, and that will produce some second messenger, usually cyclic AMP, which brings about a cellular response. The other option was that the subunit would activate a different type of channel. Okay, so there are two different options here. The subunit could activate an enzyme, which would create some second messenger, or activate an ion channel. So the important take-home message is understanding the difference between a fast response and a slow response. Okay, I want you to practice writing this out, drawing this out, describing these when you go home. ionotropic receptors versus metabotropic receptors.